So my name's Jason Nichols. I work with Syngenta and out of North Carolina. So thankfully the weather's warmer here, but it's still raining, so that seems to be following me everywhere. I apologize for that, unless you're really looking forward to rain. So I'm going to walk you through, and albeit briefly, some of the ways that Syngenta is applying the Axiom and Eureka platforms for filling a gap that we've had in low density and medium density genotyping applications. So I promise really just one corporate slide that you have to deal with, and then I'll get into more detail. But Syngenta, for those who aren't aware, is that we're a global agricultural company. We have R&D sites in over 100 countries across the globe, with many more field sites supporting that. And we have a presence in both the seeds and chemistry spheres. Our idea is to provide whatever combination of those two are required for any particular grower's needs. And we have activities in a range of crops from broadacre crops like corn and soybean, cereals, rice, vegetables, and what we call diverse field crops, or specialty crops like sugarcane, and then all the way down to lawn and garden. As with many companies that are in this area, we are liberally using genotypic data across our R&D pipelines. This is a very, very obviously generalized graph, but it's just to illustrate the point that for us, genotypic data are important from the earliest stages of research to all the way through to supporting things after they've already been commercialized, and everything in between that. And of course, generally, you can break it down into, we have a range of needs, everything from very high marker densities across a few samples to a handful of markers that we want to follow across many, many samples, and everything in between. So you can imagine that to deliver to that, we have to also support a range of technologies, everything from TaqMan, PCR, array tape, to arrays, to high throughput sequencing, et cetera. It's really, the key here is which technology is going to fit into which niche and provide the right type of data to our internal customers at the right costs for the scale they need and the turnaround times they need. So this is a very generalized view of molecular breeding at Syngenta, and it's generalized obviously because we support a lot of crops, and every crop is going to tailor this to their specific needs. But just to sort of set the stage as we get into this discussion, so there's really two big buckets that you have. So the initial upfront part is population creation, starting with parent selection, creating progeny either through double haploid, single seed descent, or other methodologies, selecting progeny, and then moving that towards performance selection. Obviously, if it's something like soybean, we just stay with inbreds, but if it's something like corn or other hybrid crops, you have a hybrid selection phase as well. And then there's several rounds of performance testing involved in the selection of ultimately those materials that we commercialize. So for all of that early stage selection, we're using performance testing data to help us apply genomic selection at several key points. So at parent selection, and then when we select progeny, as well as when we're doing hybrid selection. And as Farad mentioned, he described very nicely, I think, where what you need in a lot of these early selection phases are not an enormous amount of genotypic data, but you want to follow a relatively moderate size set of variants, but just over a large volume of samples continuously. So you want consistent data, you want robust data. And so this is a gap that we have. And because at these stages, it's really sort of a numbers game. It doesn't matter so much where your genomic predictions are coming from and how robust those are. At some point, it's just about how much material you can screen. The more material you can screen, the better chance you have of finding high-performing materials and increasing your rates of genetic gain. So what we are looking for is something that's cost-effective, medium density, or low to medium, depending upon how you define that term. I use it to describe anything from a few hundreds to a few thousands of genotypes, but to apply at those early genomic selection stages. So this getting into the platforms that we use. So again, what we need is something that's low cost, fast turnaround, so we can scale it. It's about scalability. And I'll start with on the lower density, and so I use that term to say a few hundreds to around 1,000 or so SNPs. So a few years ago, we came across, at the time, Eureka, when they were a standalone small company, and had some discussions about their technology offering. And it looked really interesting, because at the time we were also looking at, or we were really intent on that lower end of that gap need. And so they were offering something that was described as rapid, and of course everybody says their technology is rapid, so you have to take that with a little grain of salt until you do it yourself. But targeted, flexible, and affordable. So those are the 4 terms that are always going to get your attention. The other key value add that isn't really captured here is they were offering it as an end-to-end surface. So DNA to data, for instance, and that was key for us because there's a lot of platforms out there. There's a lot of technologies, but not a lot of technologies where there's also a service offering. So that's something we were interested in. The technology platform itself was actually fairly simple, fairly straightforward. For any one sample, it's only about a 2-day turnaround time. Of course, the overall turnaround time incorporates a lot more than just that particular step. But the output is through NGS reads instead of fluorescent probes, so it's a targeted GBS. But the nice part for us is the data still is scored using automated scoring algorithms as scatter plots. So this is something that's just very familiar with us. It's what we apply to other platforms. And that makes it easier for us to integrate such a technology when it is aligning with some of our practices from other platforms. So just as a very brief discussion of how we go about developing a panel for Eureka. So most of the markers that we utilize, as you can imagine, are what we would call anonymous markers, or markers that are highly polymorphic and fairly evenly spread around the genome. So these are coming from other high-density arrays, for instance, or genotyping by sequencing, more random shotgun sequencing, et cetera. But these are markers that we've just selected for a fixed panel that are robust enough and polymorphic to use for line discrimination. Also, we have to have flexibility for must-haves, something like disease resistance or whatever other traits are of high importance for target markets for that particular crop. Usually these are relatively a small number compared to the anonymous markers, but still these are ones where our breeders are asking, I need these on my array. So generally what we find is that with Eureka, the design rate is pretty good. The way Eureka is set up, it comes out as a blend of oligos. So there's an opportunity to optimize troublesome assays along the way, and it's especially important for some of these must-have assays, which are not always the most conducive to assay design, so we put a lot of extra effort in there. So that's valuable to us. And in this particular example, we saw about a 78% return on the assays that we started with, coming out through validation and final panel development. And that's generally, that's going to vary by crop. Some crops are going to be more difficult than others. But so we generally take the approach of we'll over-design, design against 20 or 30% more or more depending upon the crop, and then just reduce down to the most informative that come out of the panel development. But still, some assays are always going to be problematic. We have plenty of assays that you either have ambiguous nucleotides or some sort of variation close to the target SNP. That just creates difficulty. And while we have the option with Eureka to spend additional time and effort trying to design against those, some assays are just not worth keeping. So we end up dropping those. And with Eureka, the probe position is fixed, so that does limit a little bit what we can do with those challenging footprints. But again, with the exception of the must-haves, most of the time we just over-design and then reduce down to ones that are a little bit more amenable. One point of vocabulary, so I tend to just stick to the word SNPs, but mostly I think of that as umbrella for SNPs and small indels. So we treat them basically the same. So one of the values that or I'd say one of the key values we realized with Eureka is it's allowed us to run sufficient volumes that we are able to actually move up our genomic selection to an earlier stage. So, in an even more generalized view of the initial stages of seed breeding, so if you're applying genomic selection at this F subscript n, we're able to move that up one step to where we're applying progeny selection at a stage earlier, really before we're actually having to do field trials. So this is about allowing us to select material for field trials that's more promising and focus those resources. So that is a huge value add for us. And the other aspect of Eureka that we really like is it's, again, because it's a blend of oligos, you have the flexibility to add or remove assays as you go along. As new assays come along that are more informative, or if you realize that some of your assays are just no longer informative, it's easy to just create a new blend that drops some, adds some others. And the benefit there is you just don't have to revalidate every single assay. You have to revalidate the blend, and you have to, of course, spend the time on those new assays. But you save some design cost in those redesigns. So that's valuable. Still, as with any technology, there's a whole set of obstacles that come as you move towards implementing something into production. First and foremost is adapting to a production workflow. And as Syngenta is a big company, and we've been doing genotyping for quite a while, we have an existing production workflow that this technologies such as this need to integrate into. So it's everything from we have to have the right approaches to manage samples, as well as our DNA preparation methodologies. How do we get samples to the services lab in the right way and manage that sort of process? What's the data look like? What's the right format? And what's the right format for ingestion of the data into our systems? And then ultimately, how do we QC the data? And then what's the optimal way to get that to our customer? Underlying all this is the need to have robust track and trace and robust practices to manage capacity. For Eureka, as well as I'll mention soon in Axiom, we can reach peak volumes of tens of thousands of samples per week in some cases. So I don't want to underestimate the effort that we spent, certainly, at implementing those practices to make sure we were comfortable putting that sample volume through. The other aspect, again, I'd say this is true with any platform, is atypical clusters certainly come up. It's one thing if you have a handful of assays, you can spend a lot of time looking at scatter plots. Once you start getting into a few hundreds, a few thousands, you're going to go blind by looking at scatter plots. And no matter how important an assay is, there are just some assays that come out, they cluster atypically. And I'll get into some reasons why and some of the things we're doing to mitigate that, but that's just something that we also spend a lot of time on for new panels and just getting used to the process you have to go through to optimize those as we implement such a technology. So Eureka is a great fit so far for us for the lower end of our needs, so that's a few hundreds to 1,000 or so variants. But we also have needs that are for a few thousand, or in some cases even more, assays for crops like corn, where you have lower LD, you have a lot more genetic diversity. You just need something that has a bit larger set of variants to go along with. So a key benefit of actually working with Eureka is that at the same services laboratory, the Axiom platform was also available. So one service provider, two technologies. So that's a lot of simplicity in how we can manage things. And as we were discussing this particular need, the use of the Axiom 384HT platform came up, so we started to look at that. And that's something we've implemented for a lot of crops as well. It's contrary to Eureka, it's not an oligo blend, it's a printed array. It uses photolithography to print it. So that does reduce your ability to add or remove assays over time without just having to redesign the array. However, one of the biggest benefits of this is illustrated in this figure that I borrowed from their website. But it's the ability to multiplex crops. Because you're printing the probes on an array, you know where the probes are located. So you can, as long as you have space on the array, you can print multiple crops. This is something we do routinely. And this is especially valuable for our smaller crops, which have more limited budgets and struggle sometimes to provide, or may struggle to foot the design bill for an array all on their own, or justify it with their sample volumes. So they can add their probes on with other crops and share some of those design costs. Axiom also has a lot of flexibility in array design, similar to Eureka. Typically, we're mostly just designing to either the forward strand or the reverse strand, and just based upon an in silico analysis of which is the more favorable strand. But if for more complex or quote-unquote must-have assays, we will often or usually develop probe sets just for both directions to be safe, and then add additional replication on the array. And again, depending upon how much space you've left yourself on the array, we can even design, or oftentimes we will design probes for footprints that may have a lot of polymorphisms in them. So we need additional probes to capture all those different permutations. But we can do that, and we can query the probe sets independently, and then use the data to select which is the best probe set. And oftentimes we found that that will depend on the different populations. We do also work with larger structural variants or also GM assays. We handle them the same way. So certainly can approach this with the array platform. Just have to, we manage the design a little bit differently to where obviously you can't have both alleles, the probes for both alleles at the same location. You have different locations for the insert versus the absence, so across junctions or to other areas in the genome to control for deletion. So the same issues or the same obstacles arise for implementation for Axiom as there is for Eureka. I did want to talk a little bit on the latter part of that about the challenges we sometimes have with cluster quality and how we go about mitigating that. So there's a lot of factors that contribute to that, as you can imagine, and most of them are obvious. Footprint quality, right? That's a key thing. If you don't have good footprints, you're not going to have good assays. One of the things that we really like is that both platforms accept in the design phase our footprints. So there's no requirement that we submit coordinates to a reference genome as we found with other platforms. And this is actually really, it can make things more complicated on one hand, but it's actually pretty valuable for us because a lot of the times either the reference genome is too far away from our breeding material, or we have footprints that we've developed from internal proprietary reference genomes. So we have a lot of different rationale for using our own footprints. So that's a key value add for us. But still, using our own footprints comes with its challenges because often your footprints have some issues. And we find a lot of times we have flanking variants. And for something like corn, for instance, which has a SNP every 28 or 30 base pairs on average, this can be a huge problem. There's a paper that came out, I think in 2012, that talked about how you can take account of this, for instance, and what you see is a lot of this flanking variation will come out as as another cluster that's labeled in blue here. It's called an off-target variant. We see this a lot with corn. And so this is manageable. You just have to note it and make sure that with the automated genotyping workflow or analysis tools, you can apply an OTV caller that can help capture that. The biggest challenge is, especially if you don't have a normal het cluster in there, this can be miscalled as a het. So if you don't take that into account, it can create challenges. But it's something you can mitigate during the A, based upon the crop. Is that something you need to just be aware of just based upon the nature of the crop like corn? And B, as you're going through validation, if you're seeing this third cluster or the fourth cluster in a place where you're not expecting one, then you just have to go back and ask, what is that? Is that a true het? Is that something else? DNA quantity is, of course, important, as with anything. If you don't have DNA, you're not going to get genotypes. But what we actually found in our own hands is that the thresholds, both for Eureka and Axiom, are pretty conservative. And we don't really find too much of a drop-off in data quality until we get to diminishingly low amounts of DNA, so in the single digits nanogram per microliter. So it is important, but it doesn't always correlate with success failure. What we find more often than not is it's DNA quality or sample quality that comes into play. And we see this mostly in impacts to the het cluster. You see more diffuse het clusters, or what's been described as walking hets. So this comes into play in some applications where we use either samples that are bulked or perhaps heterogeneous tissues like seeds. So again, it's just something that needs to be accounted for in your workflows. One of the things that we do to help mitigate that is we take liberal use of priors for scoring, and we've done this more and more as we've gone along, as we realize this is a valuable thing to do, not just for troublesome assays, but for as many assays as possible. This is especially important in those cases where to date you've only seen 2 clusters or maybe even 1. Maybe you have a monomorphic assay that's still really important. The use of priors can help mitigate any shifting genotype calls across experiments, as you can see here. The other thing that we do is try to take account of population structure. On this graph on the left, in this particular case, just is illustrative of what we see. As Farad noted, is what you can see is a drop in call rate if suddenly you have material in your experiment that doesn't quite represent where your scoring algorithms were developed against. And so you can see a drop-off in call rate, and this is often manifested initially as a drop-off in the number of samples that make it through the QC steps that are dependent upon call rate. So first, key point is that one of those metrics, and this is a little bit specific to Axiom in the way this is applied, but the same concept is applied. We look at for Eureka. The probe sets that are used to define a QC call rate, as it's called, are derived from really high-performing assays or poly-high-resolution assays that come out of the validation. So if suddenly you're presented with material that wasn't really adequately represented in your validation, or if it's just divergent or small subset of your experiment, those probe sets might not be appropriate anymore. And what you see is a drop-off in overall call rate, but also a drop-off in that QC call rate, so you get a lot of additional failed samples. Another way to experiment I'll show you that illustrates that in this one, we had a plate that came back where the majority of the samples, roughly 60%, were below the QC call rate metric for pass-fail. So another way of saying that is for this experiment, 60% of the samples came back as failed. We didn't have genotypes, which, as you can imagine, our genetic leads get a little bit antsy if 60% of their data doesn't come back. So when we looked into this, it turned out that this represented some subpopulation structure that we provided information for. We then, with the folks at the Research Services Laboratory, then reselected probe sets for QC that were based on that subpopulation, and then also just rescored that subpopulation by itself. And lo and behold, you get 95% that pass QC, and you get really good data out of it. So it just illustrates the idea that the more information, what we found is the more information we are able to provide with our experiments in terms of expected population distribution, the better that can be accounted for in the data analysis workflow. And this is all done through the services, the end-to-end service that's applied. So that's really valuable as well. Some opportunities that we are exploring and further developing. These are things that we're already doing, but trying to develop a bit more. So structural variants are important, of course. We already have an interest in copy number variation, but so far we've mostly limited our efforts to just calling low, medium, high. But as we get more into this, we have an interest in pushing into higher resolution. So instead of low, medium, high, maybe a 2-copy versus a 4-copy for traits like a disease or something like that, where allele dosage is obviously a key factor in resistance versus susceptibility. Also just optimizing how we define or design around larger structural variants, dominant markers, as Farad mentioned. We obviously don't just limit our genotyping to leaf tissue. We do a lot of genotyping on seeds as well. And that's just a matter of accounting for some of the complexities that seeds bring. There's compositional issues that can potentially influence the quality of your DNA sample. So starches, oils, polyphenols, et cetera. These are things that can either inhibit some of the PCR steps or hybridization steps. The other aspect is things like, the presence, how significant of an issue might be a maternal seed coat or mixed ploidy in the seed. Something like corn with its very large endosperm, triploid endosperm could give you something like this on the right with spread het clusters. So none of these are insurmountable. These are just things as we apply each platform to a seed tissue, just things that we have to take into account and make sure we're managing on our validation side. So I'm going to wrap it up there. Just a few key points. Both platforms we're using in production for our early genomic selection. And as I said, it's allowed us to improve volumes enough to where we're starting to be able to modify our where we're applying genomic selection. So there's some return on value there. Don't underestimate certainly the effort and time it takes to take something that's small scale and make it available for very high peak volumes. As I said, we can approach tens of thousands of samples per week in some cases for some crops. So it takes a lot of time and effort and patience to get something up to speed and integrate it into our production workflows for that. Both platforms have pretty quick turnaround times. We're pretty happy with those. Eureka, especially with its ability to scale to higher volumes and with shorter turnaround times than Axiom. We are also looking at that for more demanding applications where cost and turnaround time are even more strict in terms of those requirements. But that's to be determined.